Hello again, and thanks for tuning in to Mr. Ulrich's Land of Biology.com. I am Mr. Ulrich. In this short video, we're going to look at some more complex patterns of inheritance. We've already looked at some of the more basic concepts that Gregor Mendel has gone over. These patterns are a little bit different, and they're going to start to fold in some of the more modern theories and understandings of genetics. In the last video, we talked about Gregor Mendel and how he was able to come up with basic laws of genetics without knowing anything about genes themselves and how they work. And you might remember that I kind of put those quotey marks around the word laws because though they hold in concept basically, when we start to think about them with modern understandings of genetics, they don't always hold water. So in some ways Mendel was lucky in the traits that he chose. Most of the traits that he chose were controlled by one set of alleles. We call these monogenetic traits. We know now that most traits that we're going to talk about are actually controlled by more than one set of alleles, or polygenetic traits. So he was kind of lucky that when he talked about round versus wrinkled peas, or yellow versus green pods, or inflated or shrunken pods, all those traits that he was talking about, they were, for all intents and purposes, monogenetic traits. If we're going to continue to fold in modern genetics, what we understand about genes that Mendel didn't know about, onto Mendel's laws and onto these complex patterns of inheritance, we really got to talk about dominance here. So we talked about what genes actually code for, and of course, hopefully you're saying that they code for proteins, and you'd be right most of the time. See, it gets a little bit more complex when we start talking about dominance and recessiveness. A lot of people think that dominance means like, oh, the brown-eyed allele is going to beat up on the blue-eyed allele, like it's bigger and stronger or something like that, and that's not really the case at all. For instance, let's take the example of blue-eyedness. If we go far enough back in history, we're going to get to a point where there wasn't anyone with blue eyes. There weren't any blue-eyed people around. And that first person who was born with blue eyes probably had a mutation that occurred in the gene that coded for a protein that caused melanin to be produced in the iris. And that would make brown eyes. But this person had a mutation in that gene, and so the gene was broken, didn't actually make that protein, and what ended up happening, of course, was without the protein, the melanin isn't produced, and the iris, instead of being brown, is the blueness of the collagen that's kind of left behind. So this trait of blue-eyedness is actually due to the lack of a particular protein, and that's what we call a recessive trait. And that's how you can have in a heterozygous individual who's have a dominant brown allele and a recessive blue allele, how that person will show the dominant trait because they have an allele that codes for a working protein. And that's all it takes in order to have brown eyes be expressed. So that blue-eyed allele is actually a broken gene that no longer codes for a working protein. And that's going to be the case with recessive alleles. So what does this mean for Mendel's laws? Not all alleles are dichotomies. It's not an either-or situation. It's not like brown eyes versus blue eyes in all cases, though many of them are going to be like that. Not all of them are going to be just straight-up dominance versus recessiveness, or that whole protein versus no protein situation. Different alleles can code for different proteins that influence the same characteristic as well. Incomplete dominance is an example of one way that you can have two different alleles that both code for a working protein. This way you're not going to actually have a recessive allele. So this is going to cause some interesting changes in expression. Incomplete dominance is one of three patterns of complex inheritance that we're going to talk about here. The classic example that people talk about when we talk about incomplete dominance is usually flower color in 4 o'clock flowers. With incomplete dominance, because there's no recessive allele, there are 2 alleles, but because there are 3 different combinations that are possible of those 2 alleles, we're going to have 3 phenotypes, rather than in dominance and recessiveness, a straight up protein, no protein type of situation. You have 2 phenotypes for the 3 different genotypes. So you can be homozygous dominant white, and in that case you'd have the white flowers, or homozygous dominant red, which in case you'd have the red flowers, or you can be heterozygous red white, where you'll have the pink flowers. I always think of this as kind of like mixing paint. So neither protein or neither color is fully expressed. It's neither red nor white. It's some intermediate color. Codominance is another pattern of inheritance where there is no recessive allele, and we have 2 different alleles that both code for a different working protein. The classic example of this is in coat color in different breeds of horses. We'll also see this in some cows. In this case, just like with incomplete dominance, where there were also 2 working alleles, in this case there are 2 working alleles. There are 3 different combinations that are possible, so it's 3 different genotypes, and each genotype has its own particular phenotype. In the example I'll tell you about, you can either be homozygous black or homozygous white, or you can be heterozygous. And in this case, instead of mixing paint, I think of this more like mixing different colored marbles. In this case, with the roan horse, the heterozygous black-white, each individual hair is fully black or fully white. So there are both black and white hairs present. So both of those traits are fully expressed. There isn't like an intermediate color until we back up and, due to resolution, the lack of our resolution, to tell individual hairs apart, or I smear them together into this neat pattern called roan. The last complex pattern of inheritance that we'll look at in this video is called multiple alleles. The classic trait that's given as the example for this pattern of inheritance is the ABO blood typing system. If you need to review the ABO blood typing system and what it all means and all that good stuff, go back to the video on blood, the liquid tissue, back in the Human Anatomy and Physiology unit. As we were saying, multiple alleles, for every trait that we've been talking about so far, there's only been 2 alleles possible out there in the population, right? For the roan, it was either the black or the white. For the 4 o'clock flower, it was either the red or the white. For eye color, we've always been talking about blue or brown, so it's always been 2. As the name multiple alleles kind of suggests, in this case we're talking about more than 2 alleles possible for a trait, and that's more than 2 alleles out there in the population. That doesn't mean that you or I can have more than 2 alleles for this particular trait, not without there being some chromosomal issue going on, I'm sure. So we still only have 2 of these. As we saw with the blood typing, ABO blood typing system, there are 4 different blood types, and with with multiple alleles, where there are 3 alleles possible, there are 4 phenotypes. To get a better feel for how multiple alleles plays out in the ABO blood typing system, let's look at some of the alleles that are present, or possible anyway. The capital I with a superscript A indicates the allele that codes for the A-type antigen. Just a little reminder there what antigens are, those receptor molecules on non-self cells. So we can have an A-type allele, we can also have a B-type allele that's going to code for the B antigen. In the case of multiple alleles, we got that 3rd allele. In this case, it's a recessive one. This recessive allele doesn't code for either antigen. As you'll remember, that's how recessive traits work. So let's look at what happens when we start to put these different alleles together into pairs and how that plays out as a phenotype goes. So if we're homozygous A, You have the instructions to make those A-type antigens, and you're going to be type A blood. Likewise, if you're heterozygous A, you still have the instructions to make the A-type antigen. Now, you might have fewer instructions, but you still have the instructions, so you're still going to make them. If you are homozygous B, likewise, you're going to make, you have the instructions, so you're going to make those B-type antigens. Just like if you are heterozygous B, you still have those instructions. Now, in the case of the AB, the heterozygous AB individual, they have both the instructions to make the A antigen as well as the B antigen. So in their forest of receptor molecules on the outside of their cell membranes, they're going to have both the A-type as well as the B-type antigens sailing out there, kind of like on a horse that has both black and white hairs, individual hairs, both black and white, as we were talking about with the codominance. So type AB blood is a good example of codominance. Lastly, we have the homozygous recessive individuals who lack instructions to make either the A or the B antigens, so they're going to be type O blood. If you remember back when we were talking about blood type, and I always say that it shouldn't be type O, it should be type 0. Alright, well, we'll stop there with these three different patterns of complex inheritance. I'd like to thank you for stopping in to MrUlrichsLandofBiology.com and checking out the video. If you have any feedback, please make sure that you send me an email at kulrich@newpaltz.k12.ny.us. And you can always go to MrUlrichsLandofBiology.com to get the supporting documents, notes, and other sheets and other videos on other topics. Keep tuning in. We'll see you in class.